Ever wonder how bacteria breathe? I mean, we need oxygen, right?
Right.
But some bacteria have figured out how to breathe in ways we wouldn't even think possible.
It's true.
Really is.
Take Shewanella oneidensis, or MR1 as the scientists like to call it.
Uh-huh.
This bacterium has this almost superpower-like ability.
Yeah, it can actually interact with stuff outside its own cell.
That's right.
Things it can't even eat in the traditional sense.
Yeah.
It's like snacking on metal, kind of.
Yeah.
We're going deep on this one today.
Yeah.
Looking at a paper by Korsol and Gralnick where they basically took apart Shewanella's breathing apparatus and put it back together again.
A sort of, if we break it, will it still work
Yeah.
kind of experiment.
Exactly. And as you can probably guess, they made some pretty surprising discoveries. But before we get to the mind-blowing stuff, let's start with the basics. How does Shewanella even breathe without lungs?
Well, you see, at its core, breathing is really about moving electrons around.
Like electricity, but on a teeny tiny scale.
You got it.
Okay.
And this is where the electron transport chain comes in.
Okay.
Picture it like a cellular relay race. Electrons get passed from molecule to molecule.
Right.
And this movement, this flow of energy, is what powers the cell.
Right.
Most organisms keep this relay race contained within their cells.
Makes sense.
But Shewanella does things a little differently.
It's like Shewanella decided to take that cellular relay race and extend it beyond the finish line out into the world.
A very good way to put it. And it accomplishes this with the help of a special set of proteins embedded in its outer membrane.
Okay.
Think of them like specialized ports or channels that allow Shewanella to interact with the outside world in a way that most bacteria can't.
Okay, so we're talking about some seriously cool bacterial machinery here, but this is where it gets a bit complicated. The paper calls this breathing system the MTR respiratory pathway. Got any way to make that a bit less of a mouthful?
Imagine the MTR pathway, like a specialized channel, a dry bridge that lets specific things in and out of a castle, perhaps.
Ooh, I like where this is going. So what are the key players on this drawbridge? You've piqued my curiosity.
Well, you have your paraplasmic electron carriers, or PECs, for short.
Right, got it too.
Yeah, think of them as the messengers running back and forth across the drawbridge, carrying those all-important electrons. Then you have the outer membrane cytochromes, or OMCs.
Okay.
These are like the docks on the outside of the castle walls where the electrons are actually delivered to things like metal ions.
Okay, so the PECs hand off the electron-filled package to the OMCs, which act like delivery points.
Precisely. But they can't do it alone. They need that drawbridge.
Right. I almost forgot about the bridge itself.
That's the third key player, a protein called MTRB.
Okay.
It forms a pore through the outer membrane, allowing those PECs and OMCs to interact and complete the electron handoff.
So the PECs and OMCs meet in the middle of the bridge for the exchange. That's incredible. And is this where the research takes that surprising turn you mentioned earlier?
You're catching on quickly. This is where Korsol and Gralnick really started digging into the specifics of the MTR pathway. And they found that Shewanella has multiple versions of these components. Different PECs, OMCs, even different types of MTRB.
It's like having a whole bunch of different messengers, docks, and even different styles of drawbridges. What's the point of all that variation?
That's the million-dollar question, isn't it? And that's exactly what the researchers set out to discover.
Okay, so Shewanella has all these different parts, different messengers, different docks, different drawbridges. It sounds like a recipe for chaos. How did the researchers even begin to make sense of it all?
Well, they did something really clever. They used a technique where they could systematically delete and then add back different combinations of these genes in Shewanella. Think of it like playing with molecular Lego blocks.
Okay, I love the Lego analogy. So they messed with Shewanella's genetic Lego set to see which combinations actually worked. What were they looking for specifically?
Exactly. They were testing how well Shewanella could transfer electrons to iron, kind of like a benchmark test to see how well it could breathe with its newly assembled pathways.
So they took away some Legos, added others, and tested how well Shewanella could snack on metal. Makes sense to me. What did they find?
Well, as you might expect, some Lego combinations worked better than others, and this is where those surprising discoveries we mentioned earlier come in. For example, they found that a protein called MTRE could actually step in and replace MTRB as the bridge in our drawbridge analogy.
So Shewanella has a backup bridge. That's pretty cool. But I'm guessing it wasn't quite as good as the original.
You're right about that. It worked, but not nearly as efficiently. It's like trying to cross a rickety rope bridge instead of a sturdy stone one. You might make it, but it's going to be a bumpier ride.
Okay, so some Lego swaps worked, some were less effective. What else did they find?
Well, here's where it gets really interesting. They found some components that seemed completely useless for reducing iron. Specifically, a protein called DMSF and another one mysteriously named SO4360 just didn't seem to fit in anywhere. It's like finding a couple of Lego pieces that don't seem to connect to anything else in the set.
Useless Lego pieces. That doesn't sound right. Evolution is usually pretty good at getting rid of things that don't serve a purpose. There's got to be a reason Shewanella hangs on to those extra pieces.
Yeah. You're absolutely right to be skeptical, and that's one of the big questions this research raises. It's like the researchers have stumbled upon a hidden door in Shewanella's basement, and we're dying to know what's behind it.
A hidden door. Now you're talking my language. But before we get too caught up in the mystery of those useless components, let's zoom out for a second. Why should we even care about a bacterium that can breathe on metal? That seems pretty niche, even for the deep dive.
You're right. It might seem like a weird quirk of nature at first glance, but it actually has some potentially huge implications for the real world.
Okay, I'm intrigued. Tell me more.
Well, think about it this way. Shewanella's ability to breathe on metal to move those electrons around outside its cell means we could potentially use it to clean up pollution.
Wait, really? Bacteria cleaning up our messes? I need details.
Imagine releasing these tiny metal-breathing bacteria into an environment contaminated with something like heavy metals. They could essentially breathe on the contaminants, changing their state and making them less harmful to the environment. It's like a tiny army of microscopic cleanup crews.
Wow, that's incredible. Bioremediation at its finest. And I think you mentioned something about generating electricity, too. That connected to this whole metal breathing thing?
Absolutely. Shewanella's electron shuttling abilities have caught the eye of scientists working on developing microbial fuel cells. Imagine harnessing the power of bacteria to generate electricity from waste products. We're talking about a potentially revolutionary source of sustainable energy.
Okay, now my mind is officially blown. So we've got pollution cleanup and green energy, all thanks to a bacterium that can breathe on metal. Who knew microbes could be so useful? But what about those mysterious extra parts we talked about earlier? Could those lead to even more applications down the line?
It's certainly possible. That's what's so exciting about scientific discovery. Every answer often leads to even more questions.
And sometimes those unanswered questions can be the most intriguing ones. Like, why Shewanella has those seemingly useless components. What are they really for? I have a feeling we're about to go even deeper down the rabbit hole. Okay, so we're back to those useless components of Shewanella's electron transport chain.
Yeah.
It's like finding extra buttons in a brand new shirt, you know?
Yeah.
They must be there for a reason.
Right.
But you can't for the life of you figure out what that reason could be.
I like that analogy. And in the case of Shewanella, it seems like those extra buttons might actually be incredibly important, just not for the reasons scientists initially thought.
Okay, you've got me on the edge of my seat.
Yeah.
What's the deal with those mysterious extra components?
Well, remember how those proteins, DMSF and SO4360,
Right.
they just didn't seem to play a role in reducing iron.
Right.
It was as if they were part of a completely different toolkit.
Right, like they belong to a set of galactic Legos instead of the pirate ship we were trying to build.
Exactly. And it turns out those useless proteins might not be so useless after all.
Okay.
Remember how we talked about how Shewanella has this amazing ability to interact with the outside world?
Right.
Well, it seems like it might have different ways of doing that depending on what's available in its surroundings.
You mean it's adaptable, like it can switch between different breathing modes?
You got it. It's like Shewanella can choose to breathe iron one day and then switch to something else entirely the next. Talk about having options.
Okay, that makes a lot more sense. But what else could Shewanella breathe besides metal? What's on the menu for a microbe with such a unique palette?
Well, it turns out that those seemingly useless proteins, DMSF and SO4360, they actually cluster together with genes that are involved in reducing a completely different compound called DMSO.
DMSO.
It stands for dimethyl sulfoxide, but you don't need to worry too much about the technical details. What's important is that Shewanella seems to have a separate pathway, a separate set of tools, specifically designed to breathe DMSO instead of iron.
So instead of judging the fish on its ability to climb a tree, we were just looking at the wrong tree altogether.
Precisely. Wow.
And this discovery opens up a whole new world of possibilities.
Yeah.
It suggests that Shewanella might have even more of these hidden pathways, these specialized toolkits for interacting with different things in its environment. We've only just begun to scratch the surface of what this remarkable bacterium is capable of.
It's like discovering that your old, trusty Swiss Army knife actually has a bunch of secret compartments with even more cool tools tucked away inside.
And the best part, We have no idea what those other tools might be or what amazing things Shewanella might be able to do with them. That's the beauty of science. Every answer leads to even more fascinating questions.
It makes you wonder what other secrets are lurking in the microbial world, just waiting to be uncovered, right?
Absolutely. It really puts into perspective how much we still don't know about the world around us, even at the smallest scales. Well, this deep dive into the world of Shewanella has certainly given me a whole new appreciation for the hidden wonders of bacteria. Who knew they could be so complex and adaptable?
It's a good reminder that some of the most incredible discoveries are often hiding in plain sight, just waiting for someone to take a closer look.
Well said. So to our listeners, we'll leave you with this thought. If Shewanella, this seemingly simple bacterium, can possess such an incredible ability to interact with the world around it, imagine what other surprises might be waiting for us in the vast and mysterious world of microbes.